this should be I think a pretty short lecture, so definitely feel free to ask questions. But the goal will be to sort of cover the topics that we're gonna use and like play around with in the exercises. So you'll have some handwritten exercises, and then some Colab notebooks, which will be running analysis on real neuro data sets, basically. So, all right, as a quick overview, the stuff I'm going to cover is by no means comprehensive. So we sort of chose some things that I think are fundamental and important in understanding, and definitely important in understanding for doing more complex things. So we'll just talk about matrix definitions. Lior talked about these a little bit. Doing regression of a couple different types and what it means and sort of in the context of a matrix. And then we'll talk about correlations and partial correlations sort of as they relate to these. And then I'll get into more like for modeling and analyzing single cell data, some of the stuff that we do or use. Yeah, that's just a little bit more specific to this area. Okay, so I want to come back to this slide. I'm not going to talk a lot about single cell RNA-seq because you heard a bunch from Lior. But what we're going to be looking at are some of the methods we use to sort of process and analyze these sorts of data. The term itself is sort of complicated. Exactly what you want to do can vary actually just depending on your task. But what we end up with is this thing we call a gene expression matrix or a gene count matrix, where in this case we have cells by genes. So we have, for these cells, these measurements of these genes or isoforms, transcripts, whatever it may be. So now I want to talk a little bit more about this point of what is a gene expression matrix and why sort of thinking about it as a matrix can be helpful in sort of the mathematical sense. And so you can ignore the sigmas on this slide. But here we have some matrix X, and what I think is a nice way to think about it is as a linear function. So it's doing something onto something else. And so we can just give an example where if you take this X and you apply it to, let's say, this vector that's pointing up, you can transform it in some sense, right? It's applying some map, in this case a linear map, and this is called a shearing transformation. There's like, you know, a matrix, I think if you just remove the 0, oh, you can't see, would just rotate it. So basically it's applying some transformation to this vector, and the idea is to think about it as sort of this function approach. So it's X on some B gives you some output. And in particular, these might be terms that you've heard about, like rank of a matrix. And what it sort of means is that the dimension of the image, which is basically for this X, it has some span of possible functions, possible outputs, sorry, that it could give you. Right? It can't give you everything, like all possible values of everything. So it has some like span, basically. So we can define this as sort of the dimension. We can define the dimension of this. We call this sort of the image of X or like the span of X. This is something that is good to think about. Also because being able to sort of define its dimension and its properties can tell us things about like redundancy in the matrix and different things about how we can solve the system. And so I want to just keep this in mind for when we talk about the regression approach. And generally for doing like matrix-based analyses. But yeah, but basically that this function mapping has some space of outputs that it can produce. Okay, so yeah, what I'm now, all right. What I'm now going to talk about is linear regression. And I think it's helpful to think about it in the context of doing this sort of matrix analyses where we're starting with this gene expression matrix. And I want to say something which will also be very important for doing the exercises. So usually you have some dependent variable and some independent variables you think it is, like let's say dependent on a function of. And often the way we describe this is you regress y on x. So y is the dependent, x is the independent. This is something we probably also all learned in like high school, if not before then. But I think that's also important because this actually sort of gets confused. Regressing y onto x is not the same as regressing x onto y, and we'll actually get to see a little bit of why that is the case, and you'll also get to play around with this in the exercises. But so the simplest case is that let's say you think your dependent variables, your y's, are linearly related to your independent variables. And so what does, oh yeah, and also to clarify, in this plot, usually you would say like y versus x. And so your whatever it is versus whatever is the y versus x. Yeah, and so I'm going to focus on specifically this convention for writing regression, although the more mathematical context is actually to flip these and to have beta transpose times x. But in this case, we're going to look at it as x is a function, y is a function of x with some coefficients beta. And if you think about, let me go back actually to a couple of slides. So basically, x is like the same mapping here in this case, and we can sort of see a parallel effect here. So y is some product of this like x function, essentially. Yeah, and in this case, we're just assuming this linear like multiplication of these two matrices. So then with that, the goal becomes to find this beta, right? So the point is like, let's say this is some x is some genes, and you want to predict some other genes expression, and you want to know how it is a function of these other genes, which ones are the more important, all this sort of stuff. We can think about how we solve this in the most sort of common approach to solving this, which is least squares optimization, I guess. And we can think about it using what we just learned about the matrix and the span of the matrix. And so you have what is called here the column space of x, but this is what I was just talking about with the possible sort of xb outputs that could be possible. And you have your y vector, and essentially what you're trying to find is this beta hat, which minimizes this, hold on, I'm trying to use the perpendicular error, basically, because this is the mapping of y onto this span of x, and then your error is going to be sort of orthogonal to that, and this is what you want to minimize. And this gives us probably something that you've seen before, the least squares minimization, and something we will investigate in the exercises is the actual solution that this gives you, which is this nice formula here. But we won't delve too deeply into that. But the point is that this is looking for sort of the closest projection of y in this x space. And this, again, might give some intuition into why it is a different model if you try to do x onto y. You're looking at a different space, essentially, of outputs. So something else that is interesting to think about, especially in terms of the assumptions that go into being able to do a regression like this and using least squares, is that this solution emerges naturally from assuming like a normal distribution on the errors. So in this model, there's some error that's Gaussian distributed. I'm sure many of you have seen this before. This is just the probability, the distribution for a normal, or you can see some examples on the, hold on, here. And it's based on two parameters, mean and variance, and so you have your mean and then the spread basically based on the variance. And so what this means in the context of regression is that you're assuming there is some mx plus b true solution, and your y is like perturbed by some amount that is Gaussian distributed, and thus the probability of your y, you can plug that directly into basically the likelihood formula, which is the value minus what you think the sort of true mu is. And the reason that I show this is because then you can optimize over this. And what that means is for all your points, for all the things you've observed, you can take the product of all their, I know I've lost a mouse, the product of all their probabilities. So for each of those yi's, because in this case you're also assuming they're all independent. And then you can basically optimize the log likelihood, or the likelihood. And all I mean to say by this is that what you get out is the exact same solution, the least squares solution. This may not be surprising because you can kind of see already in the Gaussian formula, it is already dependent on the Euclidean distance of the between those 2 points. So it's maybe not surprising that this comes out to be this. But this is important because it helps sort of give you a sense of the assumptions that go into doing something like this. And so what I mean by that is in the previous slide, we can see that you assume sort of the same sigma, the same variance for all your different points. And so what that means is, okay, so I say homoscedasticity. People have different pronunciations for this and heteroscedasticity. So I apologize if this is not the way you have heard it pronounced. But yes, what is assuming is this, which is the variance is constant across all these observations. Let's say or variables is, let's say, your genes. And this is nice because it allows you to derive what you will derive in the exercises, which is that best estimator for that beta hat. But something that is important to check in the data and something that we often do normalizations for, which I will also talk about, is when you have the case where that is not the case, where for all these variables you don't actually have the same variance, and then you are in this regime of heteroscedasticity, which might require different tools or different approaches. Or stabilization and normalization of the data if you use some approach like this. And so to give sort of a example application, this is just an example of a, machine learning based tool, but the main idea is what it is trying to do is take genes that are like transcription factors or something like this and find like their sort of the coefficients on these that lead to a good model for the output of some target genes. And the point is that you want to try to learn these sort of beta coefficients that sort of best represent those relationships. And in this case, they were trying to learn some sort of gene regulatory network structure from this, so which genes influence the output of this target gene. And in this particular case, I think they filtered for transcription factors to look at that. But, yeah, I don't know if there are any questions about linear regression. But, the next thing we're going to talk about is a slightly different version of this, particularly if you have, let's say, categorical data, or for example, like a binary variable, so y is 0 or 1. And so this could be like 0, you're in, 1, you're in the cell type, 0, you're not in the cell type. And what we end up doing, because you can't quite do just a literal y equals like x beta plus something, because this is not like a continuous variable, is to instead model the probability of this event. So the probability of being y equals 1, being in the cell type, whatever the example is. And the way that we do that is we use this logistic function here and these, all right, let me do the mouse again. And these parameters control sort of different properties of the output. And in this case, you can, given some input, produce a value between 0 and 1. So essentially, you get this probability value out from your model. And so in this case, what you do is the regression model is incorporated in here. So now it is not a direct relationship between P and the beta x model. It's you're using essentially this link function is what it's called. And so yeah, now instead we are modeling the odds of being in this cell type or being in whatever is the 1 condition instead of literally modeling the 0 or 1. Yes. Okay. And so what this ends up looking like, and this is something you also will get a chance to play around with in the notebooks, is you can build a likelihood model for this in the same way I talked about for the Gaussian errors and optimize over that. And so this is, yeah, so in this case, you have a variable which is like a Bernoulli variable. So this here is describing like the probabilities of the possible states this thing could be, which is only 0 and 1, so yi can only be 0, 1. And usually you see this in the form of like p times 1 minus p. To the whatever state. So if, for example, your state is a 1, it's going to be at probability p. And if it's 0, you will be 1 minus p. And in this case, though, your p is based on your model, your regression model, and this logistic length function. And so, yeah, that is how that ends up coming into play for each of those variables. And then this is a little harder to fit, usually using some sort of numerical procedure, but you optimize over all of your observations and hopefully get out this model that is dependent on the x. And so, yes, all right. Cool, okay, so I'm now gonna switch a little bit into talking about the covariance and correlation of random variables, but I don't know if anybody has any questions on the previous slides or like situations where they have used these methods, but yeah, you can ask. All right. Well then, so there are other ways of sort of looking at the relationships between your x and y variables. So let's say in this case you're looking at two genes and like their expression across cells. You can look at their covariance. So what the covariance sort of means in this sense is how does a change in one variable affect the other? So what is the extent to which these two are dependent on each other? Although this doesn't necessarily inform a causal or like a causality between these two variables. And so this is the formula. You also will have these, I think, written on the exercises, but you can also refer to these when you're going to like explore how these different properties work out in different settings. And then the covariance of a variable with itself is the variance of that variable. However, an important point to note is one, that this is not a scale invariant metric, and I'll talk about correlation in that sense. But the other point is that if you can have variables that are, if they are independent, then their covariance will be 0. However, you can have a covariance that is 0 and have dependent variables, and the reason is because, as you can see here, we're only taking into account like linear relationships between these variables, right? So this is something, again, to consider, again, with any metrics that you're using on data, is like, what is the limitation? And when is this the actual appropriate metric to use? Or are there also like to use other metrics to be able to investigate, let's say, other relationships between features? Yeah, and so there are other, I guess, non-parametric tests you can use and rank-based metrics that you can use, for example, if you're interested. But yeah, okay. But so now what we have is correlation, which I'm sure also many of you are familiar with. And this is, again, how strongly two variables are related and scale. So basically what you have is a scaling factor here in the denominator. And so you will get a value between -1 and 1. Unlike covariance, which can basically be anything, I think -infinity to infinity. Yeah, but this inherits the same limitations as the covariance, given that it is also in the formula, in terms of representing sort of linear relationships between variables. And that's something that's important to keep in mind. All right. So, yes, okay. So the next method that I'm going to talk about is partial correlation. And the reason that this is like a nice method to know about is one, that it's not actually that popular in terms of people using it, but it's something that's good to know about, and it sort of brings together what we've just been talking about with the regression analysis and correlation and covariance. And so essentially what this is like, let's say you have 3 variables, and this can be done for greater number of dependent variables, but you have some variables A, B, C, which could be not necessarily in neuroscience, but it could be like, let's say, the age, income, people, and education level, or something like that. And you want to know basically how 2 of those things relate to each other or correlate to each other, but you want to remove the effect of, let's say, some third variable, which, yeah, so maybe you want to know the relationship between age and income, but you want to control essentially for the effect of education or something like this. And so essentially what this amounts to doing is a regression between, let's say this is education and age, and then this is again education and what was the other thing that I said? Income, yeah, thank you. And what you do is then you can take the residuals of those models, again, where you're regressing B onto A and C onto A, and then look at the correlation of those residuals. And that is sort of a nice way of controlling for a third variable. And again, correlation, unlike sort of the regression sense, is like the correlation of C and B is the same as B and C, but that doesn't work the same way for the regression. Yeah. And so, yes, exactly. So this is a nice thing, for example, that you'll be exploring in terms of looking at gene expression and controlling for things like sex or, I don't know, other variables that might be of interest in, in cell types and sex and, let's say, maybe behavioral state, something like that you might be interested in. And the procedure, so this is the actual, and again, this would probably be useful for the actual exercises, but this is how you would calculate, for example, the partial correlation of B and C on A or against A. Yes. And this can also be extended to other random variables, so you don't need to have just A. You can have like multiple random variables that you're looking at. So I think that is largely the sort of fundamental stuff that I'm going to be talking about. I mean, so I'm going to be talking about next, like single cell data stuff, that the different ways we model the data, the different ways we do normalization and stabilization and preprocessing. But at this point, again, I also want to ask if people have any questions about the previous methods or metrics, or I guess metrics that they might use or be interested in. All right. Well, so this then is something that sort of came up during the discussion, and it's something that's actually very important to how we treat single cell data. And this is the fact that the data type we are working with, and this is obviously not the case, and it really depends on the data set that you're working with, is very sparse, it's very discrete, and it's very noisy. And so the sparsity, what I mean is you get a lot of zero counts. We were just talking about why there's a lot of like, I guess dropout, but that term is also used a little, is used a lot. And so we also are dealing with actual discrete counts. So we have these UMIs, and I'll go into a little bit more discussion of what they are and how they work, but the idea is we're getting counts of molecules. And so we're in the space of like non-negative integers, essentially, which is actually quite different than a lot of data where we make these sort of Gaussian assumptions and continuity assumptions about what we're working with. And also, there is a lot of noise. And then this is something we've also talked about with technical versus biological noise. There's a lot of sources of intrinsic and extrinsic noise, which may be important depending on what it is you're trying to model. And so these pictures are from actually Long Kai's from Caltech paper from, well, not at the time, but from 2006. And basically what they're looking at here is over time here, the production of beta-galactosidase, I think, and it happens basically in bursts, was what they were showing here. And in these bursts, the number of molecules produced follows some distribution, a geometric distribution in this case. And so what that means is that there is inherent stochasticity within these cells. So even the same gene is getting produced at different amounts, and this is why, among many other reasons, you get variability between cells, even in like a cell line, even in a controlled condition, basically. And then there are also many samples or many sources of extrinsic noise. So there's sampling during sequencing. There's, as we talked about, capturing molecules and lysis procedures and all these sorts of things that can then lead to those molecules that came from this noisy situation being sampled at different rates, for example. So, all right. Okay. So yeah, what this comes down to then is looking at these gene count matrices as count data. So these non-negative integers 0, 1, 2. And then again, I'll talk about how we derive them from UMIs. And so what I'm going to go through is, how we get to these counts from UMIs, but then also what the types of distributions are that we usually model this data with, because unless you're in sort of a very high count regime, it often makes sense to use distributions that are explicitly modeling the discrete data rather than making assumptions about large numbers and continuity, basically, of the means of those distributions. Yeah. Okay, so briefly, I will go through how these UMIs work for anybody who's not familiar. And the idea is that you have these, RNA for, in this case, different transcripts. that you have from your cell that's like, let's say, been captured in a droplet or something like that. And there's 3 of each in this case. And you have these unique oligo sequences essentially. And depending on the protocol you use, there's different ways that these get attached to the RNA. So I won't go into those details. But basically, these get attached to these different molecules during, like, let's say, cDNA synthesis. And then you do a PCR amplification step, and this is so that you can sort of measure the signal. But what that can lead to is possible bias and amplification, right? So it looks like this guy just got amplified less than the other one for what various reasons. Could be sequence dependent, could not be. But what you want to be able to do is collapse those counts. And so what you see here is that there were 3 unique UMIs that were attached to these transcripts. And in this case, there were also 3. So the end count, the UMI collapsed counts, end up being 3 and 3, which is what you would want to see. And so there are some technical details here, but one thing I want to point out is that in the literature, this can be quite confusing because people use a lot of words interchangeably. And so read counts is not quite the same as UMI counts. Usually we're operating on UMI counts actually at the end when you do filtering for cells that had enough UMIs and you have counts that have been UMI collapsed. So usually you are working at the UMI count level, but read counts would refer to the DNA, like the reads that you got from this data. And this is something that's sort of important to keep in mind because it's important to know which thing you are normalizing on depending on what your raw matrix is to start out with. But yes, that terminology can be confusing. Okay, so now we have this count data, at least that we're starting with. And again, so at this point, no normalization besides sort of UMI collapsing has been done, or no processing besides that in terms of the counts. And so what we have here is just, I'm not going to go through all of them, but basically just to keep in mind, there are a bunch of distributions that are really useful for modeling count data in particular. And the gamma distribution is actually continuous, and so I will describe why that is actually a useful distribution for us in the next slide. But yeah. So, all right, the Poisson distribution. So this is the Wikipedia page for this, but essentially what this is saying, and this is something I think Leo talked about in his introduction slides, was that this represents sort of the probability of some number of events k happening with some rate lambda, and usually rate is like per time already, so per that time interval. And so in this case, this is over the natural numbers, and so you can use this to model sort of the number of molecules that are produced at some rate, and you can probably fit that rate to the data. And in this case, so essentially the Poisson is like a one parameter model, the lambda, which is the mean, which is also the same as the variance. And so that is important to note because in that sense, that can sort of limit the distribution or may not actually be able to capture, for example, the variability or the noise in your data. And this is, again, something that we're going to look into, how to fit the data in this case, and also how to assess if this actually is the sort of appropriate model for your data, at least compared to some other distributions. So the other distribution that I'm going to talk about very briefly is the negative binomial. And so I'm going to first start with the more like generic definition, and then I'm going to talk about how it sort of relates to why it sort of makes sense for the biological setting that we're in. And so technically, what this means is it will give you sort of the expected number of, let's say, R failures until a success, which happens with probability P. So currently, those are the two parameters, R and P. And so if we want to put this in sort of the biological sense, I guess one could think about it as a success is like the burst of a gene expression ending. And so R is in the number of transcripts that were produced until that burst end happened. But it's not quite the most natural way, I guess, to think about this distribution and also to think about how we sort of got to using it. And Another way that's sort of nice to think about, and this is sort of in the trend of like multiple things leading to the same solution, is that the negative binomial can be written as a Poisson distribution where the rate parameter is not deterministic and that it's actually distributed according to a gamma. So this is the continuous distribution that I was talking about. And the reason that this has been useful and very popular in single cell is that what it does is it gives the rate parameter is now a sort of random variable. It has noise in the rate parameter, and this gives us greater flexibility. In fact, it's a two-parameter model, which also gives greater flexibility compared to the one-parameter model. I also was going to say if we talked about also in the discussion like how to sort of model data, how to look at things, how to sort of distinguish between technical and biological noise. And one of the ways that is nice is to try to model things from sort of the ground up. From sort of the chemical kinetic equations, if you will. And what is nice, and I think Lior may have mentioned this, is that the negative binomial sort of naturally arises as a steady state solution to a very basic and well-studied transcriptional system that has bursty gene expression. And so if that's something that you're interested in or learning more about, I also recommend for those who are students looking into the gene circuits class that Justin Boysenell was teach, which is BE 150, I think, something like that, where they go into much greater depth about like stochastic modeling of gene expression. And so I think if that level of modeling is something that is interesting, yeah, I'd suggest that. Okay. So yeah, the reason that we sort of moved from using the Poisson to model a lot of data sets to the negative binomial is something that you will investigate, but I can basically give you a teaser of why, which Lior also talked about, but you can sort of look at these parameters and you can reparameterize the negative binomial to be in terms of a mean and dispersion instead of the R and P if you want, which is what we do here. But you can see this relationship again where you see this, basically you see this over dispersion trend is what the idea is. And so what is useful is to use a model where the mean is not the same as the variance, where you can model the sort of over dispersion and distribution of variance essentially. And so this is something we're going to investigate in a couple of data sets, and also how to compare fitting a negative binomial and fitting a Poisson. And what's nice about that is because the Poisson is basically a sort of sub-model of the negative binomial, you can actually compare them using a likelihood ratio test, and we'll get into that in the exercises. Yes. Okay. Right, so that's all I was going to talk about with the count distribution. So now I'm going to talk about some of the normalization stuff we do, variance stabilization, because of the different assumptions that the methods make. Are there any questions about any of those topics?
Using the Poisson negative binomial, do you assume that events, do you have to assume always that events are independent, or can you use them even if there's correlation between previous events?
I guess, let's see. Yeah, usually.
So here on this graph, every point there is a realization of a negative binomial, or the entirety of it is fitted to like a histogram?
Each of these is a gene, with the mean and variance. So the mean and variance are not from a fit, actually. They are from, so you're fitting, I guess, one model to all of these points in that case, because this is just showing.
Every point is a realization from a distribution with mean and variance.
Yes, exactly. So like your gene, based on the observations, which are your cells, has some mean and variance because it has some set of observations.
So therefore, they're random variables.
Yep. So each gene is
independent from one another?
Currently, we're treating them as independent. You can yes.
Like in general, not for this example, but can you use, or do you know examples of when these distributions have been used for non-independent or correlated events?
I guess. Let's see if people want to. If you want to do well, so I guess You can model joint models. So you can model, like, for example, joint models of like a negative binomial where like the variables are correlated, but it is a little harder to fit, for example, than in like a Gaussian distribution or something like that. So they're usually more difficult to fit if you have some correlation structure between. But yes. And yes, okay. And so, yeah, all right. So then for actually using this count data, we need to look at the assumptions, I guess, the analysis methods we're using are making, and then determine if the data in its current state, will work for that, or if that method actually makes sense. Okay, so this is something we also talked about. This is something I mentioned with the least squares. A lot of these methods, like least squares and PCA, make assumptions like equivariance and I guess independence between each of your genes. Similarly, clustering, I think, which is a topic that's very common for people to do, relies on calculating distances between your genes. And so the problem with this can be that if you have a highly expressed gene that has a higher variance, that's going to contribute more to these calculations, even if that's not necessarily what you want. So there is a process called variance stabilization that you can do to your data. And the idea is to stabilize your data such that the variance is no longer, let's say, dependent on the mean. And so if something has a large mean, the variance is still sort of constant, essentially, is what it does. Yeah. And so an example of that transformation is, well, derived here, and sort of a simple way to approximate such a transformation on your data is the log. And this looks similar to probably something a lot of people have used, which is the log 1p, or just logging your data or something like this. Yeah, and so the reason is because if you log the data, you basically remove this variance equals mean whatever parameter or relationship, and you have now the variance is just constant across. And so this is not necessarily a transformation you would want to do if you're trying to look at, let's say, a difference in variable or looking for variable genes, these sorts of things. But it might be useful depending on, you just say you're trying to fit a regression model or something like that, and you're using like least squares or something as your approach. But again, this is something that, every sort of transformation has its downside. And this is something also I think Leora can comment on more if you have questions, but so issues with the log 1p and using the pseudo count in the log of x plus 1, especially when a lot of your data is zeros. But yes, so we will explore also how these transformations affect your data, and also how they are more or less effective depending on how small or large your counts are. In fact, a lot of these transformations tend to be less effective if you have very small counts. Yeah, and so there are other normalizations that people tend to do on data. And some of these are very common, especially before you do, let's say, differential expression analysis, where you are looking at sort of DE between genes, which genes are differently expressed in one cell type versus another cell type. Yes, I think that's what I'm talking about here. Right? Yeah, and so what can happen is basically these different cells can be sampled differently in this whole sequencing procedure that we're doing. And so what you may have is something that was sampled more, and thus there are more reads, but that's not actually kind of an equal comparison between cells. And so there are a lot of different things that we do to sort of try to normalize for that. And this is actually called quite a few things. It's often called size factor normalization or library size normalization or a bunch of things like this. This is, again, a good place where it's good to know what you're actually operating on, if it's a read count normalization or if you're operating on UMI counts, these sorts of things. Yeah, but the idea here is basically to scale the sort of total reads or UMIs that you have for a cell based on how many total it has. And I think here I just show dividing, let's say, all of its counts by its total. So it's like a relative amount that you're now comparing between cells, but often there are other transformations that people might be aware of, like CPM and TPM and these sorts of do similar things. They try to scale the reads in the cells to sort of a similar baseline, essentially. And this is something that's very common before doing differential expression analysis. Yeah. And usually, yeah. So anyways, depending on what the term is, people might be doing this on the reads of the data, or they might be doing this on the UMIs, and that's something that's important to know, especially if you're using UMI-based data. And so I think that is largely all that I had to talk about before we do the exercises. So yeah, I don't know if there are more questions, but let's kind of keep it short for the lightness.
All right, well,